Good morning. It's May 15th, and we've been talking about the nitrogen fixation root nodule lab, which is pretty hard to say. But essentially, we're on day 2, and we've already covered a lot of these slides in the presentation. So I'm going to scroll through here. We're going to talk about what we need to do to wrap up day 2 of the nitrogen fixation lab. So you've downloaded the lab. You've gone to you've opened it and you've read the pre-lab reading portion. Just a reminder, if you haven't done that part yet, stop this video and go to the video of yesterday's lesson and start there, because without this information, you're not going to understand everything that's going on. You have completed the tutorial session with me together, and again, if you have not done that yet, stop right here and go watch the video from yesterday, from day 1 of the lab. Questions that we had to complete for day 1. I've listed those, I've shown these here. That's what it should look like when you're filling these in. Now, in the past, I've had kids answer these questions in different ways. I've had kids write, or I should say draw their pictures by hand, and then take a picture with their cell phone, click, click, and then save that and upload it. That's an easy thing to do. I've had some kids who are crafty with the paint program on their laptop, able to do decent drawings with that. I don't expect a Picasso. I just expect it to be complete and thorough. And those are the same thing. Complete and answering all parts of the question. All right. So I'm going to show you a couple of examples of what different students have done in the past and what I think about those. So I've organized this roughly into, these are the different students down the columns, and then the questions we looked at for day 1 are across the row. So you can see all different kinds of examples that I thought were acceptable. In the first part, they've drawn this with a computer program, an art, like a paint program on their computer. This was the first part where they had to draw the roots and then the nodules on it. This was the beet, I should say the radish root. And then this one was just where they just had to do the nodules. They chose to do those in 2 different places. It looks like they just used the same picture and pasted it twice. That's fine. What I like about this also is, if you see here, there's words. So there's no real description. I should say labels, but there's a good description of what's going on. If you compare these two to another student who also used a paint program, I'm hoping that you can see the difference. First of all, this is not a beet plant. So this student did not get credit because, I keep saying beet, I mean radish. This student drew the wrong picture. And then if you look at them, the things are missing. First of all, there's no labeling. There's no indication of any nodules on this bean plant, and there ought to be because the bean plant has nodules. And then this isn't even the same plant. I have seen examples of kids who've drawn it on paper, uploaded it, and then added in their description, and that's perfectly acceptable. I've seen kids that have drawn it on paper and put a key. So this would be the root the bean root, and this would be the radish root. And this is what they literally saw, and they just drew the hand lens. I thought that was cute. What I had to crop off for you is they didn't just put this picture in, they also had some words to describe. I had to crop off the rest of the words. So these are all examples of student work that I've given full credit for. I only give it full credit if it's accurately drawn and if there are either labels or a description. For example, I really liked this student's work for the second question because they took the time to put in at least 2 responses to the differences, whatever they were prompted to write here, and their picture. So this student did it a different way. This student opened the document on their computer, typed in all their answers, and then printed it, and then drew in the margins, which would have been where their table was, and then scanned it in and resubmitted it. Whatever works for you, as long as it has everything that I want. Okay, so it's got to have an accurate drawing. It has to include labels or a verbal, like a word description. You can't do one and not the other if you want to get full credit. I also am assuming you understand that if I see these same words show up on your sheet, you're going to lose credit for everything, because at this point in the year, you should know how to be academically honest. All right, let's move along. So, starting with the second lesson, the second day of this lesson, I'm walking you through here in this part what you have to do to manipulate the microscope. If you look, I have updated the original Word document from your file. If you pulled it off the day one with us, you don't need to reprint it or re-download it, because the info I'm sharing with you is also right here. You can if you want. It is not a big deal. I haven't changed any of the part that you write on or fill in or draw on. So if you wanted to just, I don't know, save it again and print off this page, I literally cut and pasted it. It's exactly the same as you see here in this presentation. So I will leave that up to you. Let me focus back on, focus. That was an inadvertent pun. When you're doing the simulated lab, the virtual lab, it assumes at this point that you've already had one microscope lab, so you already know how to work a microscope. So for those of us that are coming in a little bit later, I'm going over this step in more detail for you because it may not be so obvious. So I'm going to minimize this for a small second. Actually, what I'm going to do is pull up this one. Okay. So let's see if I can't, since these are exactly the same, I'll try to toggle through these two things just to make your life a little easier. Okay, so this will be with us here on the side while I go through it over here. All right, so at this point, we are at the main, we went to the main menu. We had to go through all the steps for this lab that we did in the tutorial. So we cut the nodule off of the root here. We placed the nodule onto the cutting board. We picked up the knife, we cut the nodule, and we looked at it under the microscope, or under the hand lens. Now it's time for us to look at it under a microscope. So at this point, here's your microscope. What you don't see is that it came, when you open it, you're going to find there's a plastic cover on it. So just touch it and click it, and it'll pull it off for you. All right, how to work the microscope. Let's see. Over here it says, in order to work the microscope, remove the cover, did that. Turn the microscope on. So the microscope on button is right here. Click. And then it's going to prompt you to clean the lens. I must have used all my lens paper. So you'll have some lens paper here. You'll grab it, and then you'll just wipe the lens with your paper. You can throw it away. It says adjust the lighting. So right now, if you look at the lighting here, there's dark, there's midway, and then there's very bright. So let's move it back and forth, okay? Okay. So what we're going to do is take this slide. It says, for C, it says, place the prepared slide of a nodule with the microscope and use the coarse adjustment to find the tissue on the lowest power. So when I say lowest power, this is your eyepiece. This is what you look through at the top. It is always worth 10 times magnification. So if you just used this to look at something, you would see that 10 times bigger. Then you have these three lenses. There's 4x, there's 10x, and there's 40x. So how do you figure the magnification? You take this number that you see and you multiply it by 10. So this is, if I look at this slide with this setup, that's 40 times bigger than it was. This is 100 times bigger because 10 times 10, and this one is 400 times bigger. So what you have to do is start at the lowest, and then you have to use these two knobs here to adjust. What you need to understand, and it says here, the coarse adjustment knob raises the stage to the lens. So this is the stage, this black thing here, and you can move it around like so. Okay, these are the clips that hold the thing on. You don't need to do anything with those. But you may have to know how to move this stage around to find your specimen. These two knobs, this one here is the coarse adjustment knob. So when I click this, I have to hold it with my left mouse button, and then I can scroll up and down with my right, or move your mouse. You might want to take a minute and just to make sure you can in fact do that. Now watch the little red dot. As I do this, what you don't see very well or at all is as this moves up and down, so does the physical stage here. So this will move the physical stage very, very, very close to your eyepiece, this magnification lens. And when you're at 40, that lens sticks down a lot farther than when it does at 4. So as you're moving the stage, you're moving it closer to that. And if you're not careful, you can break the slide and it'll say, are you sure you want to keep moving? And if you say yes, you're going to crack your slide and you'll have to start all over again. Okay? So this is the coarse adjustment. Move the whole thing up and down. It's for fast adjusting. And this one is for fine adjusting. So you have to click on that and then move that later. But do that only when you get to see your specimen. So we're going to pick up the, come on, we're going to pick it up. And we're going to pick it up, pick it up, pick it up. Thank you. And we're going to put it on the slide. But be careful because right now I'm at 40, so I better readjust this. So I want it lowest. Okay, and then we're going to move up so we can see what's going on in here. I'm going to have to make this a little bigger, I think. Okay. All right, so what you need to do is find the specimen on low power before you zoom up onto high power. So let's find that. We're just looking for something that looks like there might be some shade or something to look at. Oh, here we go. And it's really can be tricky to move, so you've got to move slowly. But here you go. Here are the cells. And once you have it, you can move it around and find a good spot to look at. Well, there's a good spot. Let's look at these. Okay. So I'm at 4D times bigger. I'm going to use the fine adjustment just to tweak it and see what I can see. Okay, so when we look at this, we see some cells that are bigger than others, and that's what we're looking for. So let's take this up another notch to 10 times. Now, if you move this rough one, you might lose your sample. So let's try the fine first until it comes into focus nicely. Very cool. Grab your thing and let's move it over because I thought we saw some cool color somewhere along in here. When you zoom in more, what you need to realize is you might lose what you had in the center. Is only what's in the center is what you zoom in on. So if it was somewhere on the edge, you might have lost something. So this looks interesting. Let's look at this. Okay. So I'm gonna go to high power now, and I'm gonna do the fine adjustment again. Now the problem here is I've lost I've zoomed in too much with the fine. So I'm going to come here and I'm going to very carefully move the rough or the coarse adjustment. Okay, so now we can see. Oh, let's change this light source. Oh, sorry, I wish they didn't have these. Ugh. Okay, come on. How do you get down there? Okay, let's move this light source bigger. I mean better, brighter, and all that jazz. So let's look around. And okay, here, now we see some cool things. And you're going to be going pretty fast, moving around at the high speed. Okay, so good enough. Let's look at this one. So what you're looking for are the differences between the two kinds of cells. Some of the cells have an infection in them, which makes them swollen, for lack of a better word, swole, and some of them don't. So when you're looking at those, you have to figure out which ones are infected with rhizobium. You might say that those ones that are pink, remember, pink means that they're actively fixing nitrogen. There's some there. That's a good one. Let's leave it here. All right, so there's no pink. Oh, there we go. There's some small ones. So these here are smaller cells, and there doesn't seem to be any pink in here. But these other two, they have some pink, right? So those cells are actively fixing nitrogen. All right, so I'm at, what's my recording at this point? Okay, let's see. So then you have to sketch the bean nodule, including cells with and without rhizobium, showing what you observed with a microscope. And so that's what you do in this part here. Lab report day 2, observing. You write what you see here and you label it or you describe it. That is up to you. Do you remember how we discussed if they're infected, you're going to see them larger and you're going to see them like darker spaces. See, these have, you can tell where the infection is and where the empty space is. So you have to draw those. Now, if you want to draw them from 100, that's okay, as long as you are detailed. You can see a lot different cells when you go to 100 instead of 400. So you can see much more of the difference. See here? Okay. So again, I'm going to have to fine tune with this one. But you have to be real careful with the rough or you'll blow right past what you're looking at. Okay. So you can see the different size, the little pink, the bigger, fatter pink ones. These are a nucleus. This little black, these little dark spots, those are the nuclei. You won't see nuclei in every one of them because they might be on the other side. And the littler, whiter ones or clearer ones don't have infection yet. So the big, fat pink ones, those are the ones that have the rhizobial bacterial infection, and they're actively fixating nitrogen. And the littler white ones or clear ones, they don't have the bacteria maybe yet. Okay, so that's what you would draw, recreate that, and label it. So infected cells or rhizobial cells, these little white ones are not. They have no rhizobium. And these dots that you see here, those are nuclei. All right, and then let's look at this one now. So to answer the rest of these questions, let me just make this bigger. Hello. Okay. You're going to sketch the bean nodule. It says 2 new species of plants. You know what? Let's do this. I like this one better. Okay. So that's what we did here, and then the page down. Okay, so here's the questions for day 2 to complete. You've got to do conclusion questions, 3 of them. When you write a conclusion question, you should be thinking complete sentences and as many terms as you can think of to fit in that space. So for number 4, 2 species of plants were introduced to an area that was low in available nitrogen compounds in the soil. So the soil's not very rich in nitrogen. 1 formed nodules and 1 did not. Which species would you reasonably expect to grow more robustly? Robustly means healthfully, fast, successful. Okay, robust means good. And then that little word there, explain. I do not want a one-word answer. I will not even bother to grade it. Don't tell me the one with the nodules or the one without the nodules. You need to elaborate. This is where you show me what you know. I want to see vocab terms. I want to see an explanation. All right, number 5, how does the location of the nodules relate to the function of the nodules, i.e., think about where they're located on the plant. Why is that a good location? Oopsie. And then down. And then for this diagram, it's actually much simpler. It should be sort of what you saw in the microscope. You need to identify which one has rhizobium bacterial infection and which one doesn't. Label them and explain the obvious size difference using observations that you made from up here. Okay? I also wanted to show you some examples of students' work that I've taken in the past. I've only got 3 examples this time, but here's an example of a student who did this with the computer art program. Is it elementary, like simple drawing? Sure. I didn't expect an art gallery. But what I also again cut out that you don't see is they added words as a description. So what they might lack in pizzazz in the picture, they made up for in a healthy description with good vocabulary terms. This student drew it by hand. They identified, you can't see the dotted lines very well on here, but they identified, they labeled, okay? And then I didn't show you, they also put the description. Here's another one. This is from the student who printed everything out and then wrote on it and put it back in. All right, so that's at least the kind of work that I'm expecting from you to get full credit. Now, after you're done, submit it in the Dropbox by tonight at 11:30 or 11:59. Really, I'm just before midnight tonight. Okay, and I think, yeah. So I will go over your attendance password in class today. Thanks, guys. I will link another video for the day 1 in case you missed it.